Okay, hi. My name's Johan Hartman, and I'm here to do this talk for Laura. I apologize she couldn't be here. I've worked with Laura for the last 3 years on this project. It's called the Brain Index, and it's a... Oh, the videos are not playing. Okay. It's a permanent installation in the lobby of the Jerome L. Green Science Center on Columbia's new campus. And what it is basically a neuroscience visualization and storytelling tool. The task was to create something that would make the research that's happening in the building accessible to the public via 4-minute science narratives and also dynamic brain visualizations, which you can see at the top. So I'll talk a little bit about how this project came about, what the decisions were that we made, and who worked on it to make the science that's happening in the building accessible to the public. As you can see, it's pretty large. It's 24 by 24 feet. Those are robotic monitors that can move up and down. They're also touch screens. The project is I'm going to use 2 computers here for my notes. It's an interdisciplinary collaboration between these 3 institutes at Columbia. So the mandate was very much to do this internally and find ways to collaborate. The Zuckerman Institute was the main client, pretty much. So that's the client of the building, which I'll get into a little bit. What it is really an interdisciplinary neuroscience research lab. And then we collaborate with the School of Journalism, specifically the Brown Institute for Media Innovation, which is led by Mark Hansen, who's done a couple installation projects in New York, one in the Times building. And then the Center for Spatial Research, which is directed by Laura Kerrigan, who teaches at Columbia in the School of Architecture. And I was brought on to the project in 2014, so it lasted for about 3 years as the project lead, sort of to coordinate the design and architecture and the exhibit and so on. My background is that I had just graduated in 2013 from Columbia to get my master's in architecture. And prior to that, I'd worked at Google, so my background's a little bit of design and technology. That's the building. You can see it's still partly under construction, but it houses essentially 60 labs. So they do everything in there from computational neuroscience to behavioral experiments and so on. To the left, you can see the art school, and in the front, you can kind of see the University Forum. So this is the first building on the Manhattanville campus, which is to be completed by 2030. And the goal of the entire campus is to be accessible to the public. So every ground floor of every building will have some publicly accessible spaces where you can explore what's happening in the building. We worked also in collaboration with the Education Center. This is the space in the building. So the project was really a science communication and a science outreach project. And we worked with Kelly Ramol from the Zuckerman Institute to get a lot of the science right. I'm going to skip some of these. This is kind of just a plan of the space. So you can see it's 30 by 30 feet. It's quite a long hallway. The building is very tall. You can see there how large the lobby is. The building is kind of the brain of the new campus as well in terms of energy use. So there's several sub-levels that control how the entire energy production happens for the entire campus. It's surrounded by multiple quads that will house a cafe, restaurants, and the education center. Our overarching goal really was to create a way to communicate science in new ways, so to kind of break the existing neuroscience publication cycle, which is kind of confined to academic publishing, conferences, and so on. What we wanted to do is find a way to engage lay people with neuroscience. So that was kind of a challenging task. And we were inspired also sort of by neural networks and really initially conceived of the entire project as a series of connected exhibits. The Brain Index is the first one that got built. But what we wanted to do is create several exhibits in the space that all kind of talk to each other and that can address multiple audiences and have different features. So here you can see a rendering of the building, and there's a little fly-through that shows what the kind of initial idea was. So as you can see, we're connected to 2 publicly accessible spaces as well. So there you can see, this is the entrance from the Broadway side, looking towards campus, basically. There you can see the elevator bank, so that's where the scientists go up. And there's a chandelier called the thought bubble, a wall where you can explore sort of interdisciplinary neuroscience collaboration called NeuroPlus. And then to the side, you can see the brain index, which is the built piece. I'll just skip that. I'll get into it. So what really inspired us initially, and Laura's background very much is in data visualization, was brain models. So we looked at the history of brain models, very much inspired by Carl Schoonover's book, Portraits of the Mind, which kind of goes through the history of how the brain has been visualized and how every kind of advancement in visualization has led to new understandings. So there's several models in the system, which I'll go into. The model that initially interested us, of course, was the connectome model, which some of you may know what this model is, but really what this is kind of a highway map of the brain. So it shows how your brain regions are connected and which regions talk to each other more than others. And also, this model continually changes. So if your brain is imaged on one day, it may look one way, or the next day it'll look different. So as you kind of learn and evolve, your connectome model will change. So we were provided a model by one of the scientists' former grad students, and we converted that using MATLAB and a bunch of other algorithms to something that you can use in the brain index. So here you can see it converted to an OpenGL format that we can render. The entire models are all dynamic, so nothing is sort of canned video. It's all calculated in real time. And here you can see the connections that go through the hippocampus, which is kind of the region where your active memory takes place. The other models that we were interested in were functional models. So here you can see how the brain is subdivided we have 14 functional regions in our model. And this is something that, some scientists will argue about, and there's little controversies around where functional regions begin and others end. But we wanted to be able to address specific functional regions so that each story could be indexed to something. So you would know, this has to do with hearing or vision and so on. On the side there, you can see an fMRI model, which I'm sure many of you know what fMRI is, but that essentially is, which functional regions sort of light up due to blood flow and electrical activity as a person is in a scanner. So a lot of behavioral experiments are done this way, where you look at what stimulus causes what sort of response. And the other models are structural models. So on the left, you can see the subcortical structures that are inside the cortex. In the center and on the other side, you can see the vasculature model. So these are also models that we received from studies done at Columbia and then converted using MATLAB and Maya and a bunch of other tools to make them work in this system. The other thing that really fascinated us was, from a visualization perspective, was the neuron tracing. So this is the idea of, and we've seen this earlier with Kayal, the idea of tissue being stained and then people actually manually drawing and tracing neurons. Not so much anymore. It's mostly automated now, but to kind of reverse engineer the wiring of the brain to see how these networks work. This was a 3D printed neuron that was done at Yale in 2013. And that kind of inspired us as well. We wanted to see how far we could take the 3D printing of neurons. So these are some confocal microscopy scan sets that were provided by Randy Bruno, who works in the building. And we developed several processes to basically analyze these and get the most refined geometry out, and then created some 3D prints of neurons. Here you can see that's Renzo Piano, the architect of the building. And here's one of our neurons that we made. So it's pretty detailed. It was pretty hard to print. And we became sort of obsessed with neurons and these typologies of neurons, different species, looked at Neuromorpho, which are public data sets that contain some 80,000 tracings of neurons. So we kind of created a kind of taxonomy of neurons and exhibited some of these. These are some videos that we made. So here you can see some neurons from the lab of Randy Bruno, and these are fairly simple. The next one you'll see is pretty complex. It's one of his favorite neurons. This is actually 5 millimeters in length in a mouse brain, and it connects the thalamus, which is where all your sensory input comes in, into the neocortex, which is kind of what he's interested in studying. So we found a way to etch these in glass because we thought that was kind of a nicer way to show the fine structure of them. So here you can see them exhibited at the Design Biennial in Istanbul. This was last year. And what we did is we exhibited several of them along with video games that were programmed based on the experiments that the scientists did. So kind of finding ways to convert and translate science. And the next part, of course, is the science narratives. So this is what happens when the monitors come to rest at the bottom. And our goal was really to find ways to interview the scientists and distill this very complex research into something that somebody with no understanding of neuroscience can explore and understand in about 5 minutes. So to that end, we worked with several design firms, and it took quite a while to develop a kind of graphic language that would work in that it would be accessible, detailed, yet also nice from far away and friendly. And what we wanted to do was create a language that could vary in its level of complexity, and that would always focus on the human. Every story is really about what this research means for people. But also we wanted to make sure that we can stay accurate. So depicting really complex neural processes was obviously important. Also the depiction of animals was something that we had to really fine-tune. How do you depict genetically modified animals in ways that are not too realistic? So here you can see there's a mouse, there's hippocampus in the mouse brain, for example, on the right, and then the center is an electric fish, which one of the scientists studies, Nate Sawtelle. He studies electric fish and electrolocation and how that works in the fish brain. Here's some title slides. So each story kind of has a sort of engaging title, and there's 8 stories now that are live, and you can see they range from blood flow, which I'll go into this story a little bit, and then decision making and DNA. There's others about mouse whiskers and mouse brains and all that fun stuff. Each story also has an explore mode. So we wanted to create a way where this could work for multiple audiences. So if you really wanted to go deeper, right, each story has several screens where you can click explore and then go deeper and explore the actual artifacts that are produced in the building. So you can see some a slice of the hippocampus with some neurons rendered. In the middle, it's not animated, but that's a 3D neuron that you may remember from the video earlier that you can basically spin around. And then there's output from the experiments that we translated. And these are some of the interactive pieces in the content. So these are games that mostly I programmed to run on a just regular web front end. So these could work on any browser, of course, on the screens, could work on a phone. This is a dot motion experiment, random dot motion. It's used in neuroscience often. Here's an overview of what the stories are that we currently have in the system. There's several others planned, and there's 60 research labs in the building, so we are hoping that it will grow over time. And really, the entire brain index is really conceived of as an instrument for storytelling. So there's a content management system behind it. There's a storyteller kind of interface where you can upload graphics and create your stories. And you can also coordinate the choreography of the screens in any way you want using the system. So there can be call-outs. The screens can perform different motion patterns. The colors can be adjusted. Colors can blend over time. And the monitors can also follow what's happening on the screen underneath. Here you can see how the system was designed. So what you're seeing there is the robotic structure, which has counterweights and motors to drive the monitors up and down. This was pretty challenging to get this to fit within 1 foot. We wanted to make it as thin as possible. And then we covered it in LED tiles. So the reason we chose tiles was so that they could be replaced easily if one of them breaks. And we had these specially made so that the LEDs were potted in a gray surface, because what we wanted to avoid, a huge black box that's just kind of intimidating and scary. So we wanted it to blend into the space of the hallway, which created some challenges for the visualization. So as you can see, there's a little bit, the contrast is a little overblown, but you can see on the LEDs you really can't read contrast very well. So what we thought of as the monitors as kind of magnifying glasses that kind of zoom in and reveal other details. And then now I'm going to go into one of the stories. This is featuring the research of Elizabeth Hillman, who studies blood flow in the brain and what blood flow means. So the way this story begins, let me see. There's a kind of an intro sequence, and you see a number of different brain models. In our case, what we're showing is the cortex. And then you can see the system here is zooming in on the brain stem, which is where the blood enters the brain. So we can pull out these anatomical details and tell the camera to zoom in or hover around them. And then you were seeing the vasculature being rendered around the brainstem. And here are some of the stills from the story. So what you can see there on the far right is a kind of expanded view. So this is another piece of the kind of explore mode. So you can actually click on any term that may be confusing or hard to understand. So in this case, neuron, and you can always get more information about these very sciencey terms. Here's the first explore mode. And so if you click on explore there, you will go to that screen where you can play with the neuron and find out more about the pieces of the neuron and what they mean. And then the story really goes into how blood flow really controls all your emotions pretty much. And your brain consumes a lot of blood, so it needs a lot of nutrients. And this part talks about how blood flow is used in fMRI studies, which very much the activity that lights up in the brain really is related to where the blood is going. And then, of course, what happens when something doesn't work. So in this case, there's a blood clot, right, which is causing a stroke. So here you can see a stroke in a mouse brain. So at the top, you see electrical activity. Below it, you can see what happens to the blood flow. You can see this kind of wave of depolarization as basically neurons begin to fire in succession, and that's what that's what the stroke is. And we always, every story always comes back to the human, so we talk about what this means for humans and basically how having a healthy heart means having a healthy brain. And there you can see the profile. So that's it. Thank you. Any questions? Let me know.